Hello and welcome to the Beyond Biotech podcast number 48. I'm Jim Cornell from Labiotech, and we had yet another bank holiday on Monday. Not that I'm complaining, and there was no school on Tuesday either. The Scottish school year is quite different to the one in England. I remember taking my son to England while they were off school here in Scotland, and we got some strange looks as if to say, why isn't your child in school? Less than a month to go here before the school holidays, which is a bit scary. And I know I mention the weather a lot, but since the last podcast, it hasn't rained here. I've even watered the plants. I always figured that the worst job in the world, well, at least in Scotland, would be selling watering cans, but this week showed that we do still need one. Anyway, we're here to talk about biotech and not the weather. So this week we're talking about immune modulatory antibodies for cancer immunotherapy with BioInvent CEO Martin Welschof and Cecilia Hofvander, who is the company's Senior Director of Investor Relations. And so now we'll take a look at some of this week's news headlines that you may have missed over at Labiotech.eu. There were some interesting ones, if I do say so myself. The UK government is investing £650 million in life sciences. Molecular Partners has presented positive solid tumour data, and researchers have identified a gene expression signature to predict the progression of type 1 diabetes. Potential targets against natural killer T-cell lymphoma have been found. A pancreatic cancer microbiome discovery may give hope in the search for a treatment, and a new study revealed Alzheimer's disease inflammation protection. We had an article on 5 of the latest advancements in endometriosis research. Artificial intelligence has helped combat a superbug through the discovery of a new antibiotic, and we had an article on the challenges and opportunities facing biotechnology companies in developing countries. There are positive results for the first in human CRISPR microbial gene therapy. Lonza boosted its antibody drug conjugate portfolio with the acquisition of Synaffix, and researchers are fighting obesity through the use of gene therapy. Elevate Bio has raised $401 million for cell and gene therapies. Sanofi's drug has been shown to reduce disease activity in relapsing multiple sclerosis, and Swiss researchers say they have developed a new class of antibiotics to fight resistant bacteria. Anoka has raised $36.6 million for the large-scale development of cancer cell therapies. Avacta has acquired Chorus Bioconcept, and a new approach improves cancer treatment and minimizes side effects. Scientists have developed a probe to push for new drug discoveries. We had an article about 5 ophthalmology biotech companies that you should keep an eye on, and you can read all of these and many more at Labiotech.eu. And so it's time for this week's interview, and it's with a company that was in the news this week for positive Phase 1A data on the treatment of solid tumours. Transgene is a biotech company that designs and develops virus-based immunotherapeutics against cancer, and it has partnered with BioInvent International, a biotech company focused on the discovery and development of novel and first-in-class immune modulatory antibodies for cancer immunotherapy. And it's BioInvent that we had an opportunity to chat with about immunomodulatory antibodies and what the company is doing in the fight against cancer. And we talked with BioInvent CEO Martin Welschof and Cecilia Hofvander, who is the company's Senior Director of Investor Relations.
So I'm Cecilia Hofvander. I'm heading the Investor Relations at BioInvent since a little bit more than 2 years now. I'm a scientist by training, but that was way back. So I spent the last 20 plus years doing investor relations for different publicly listed biotech companies here in the area, actually in Sweden.
Yeah, and I'm Martin Welschof, the CEO. My background is also scientist by training. So I did my PhD and postdoc at the German Cancer Research Center. So, I'm originally from Germany. But I left Germany now, I think a little bit 20 years ago, but didn't go back. So I'm now living in Oslo, Norway. in the life science industry now for 25 plus years. Various places in Europe, mainly Scandinavia, but I also spent actually 7 years in Dublin, Ireland, and also worked in the San Francisco Bay Area for some time. That was also quite interesting. And this is now my third CEO position.
So could you tell me a bit more about the company?
Absolutely. So joined BioInvent in August 2018. And the reason for joining at that time was actually, there were a number of reasons, but I will just focus on the most important ones that really made this, to me, a very, very interesting opportunity. Number one, it's a relatively integrated company, at least for European standards, which means we do target discovery, we do compound discovery, which in our case is human monoclonal antibodies. We can produce ourselves, so we can generate cell lines, we can produce up to 1,000 liter GMP in-house, and then we have clinical development. So we can really accelerate very quickly once we have an interesting mode of action around a new target that we have discovered, get that quickly into the clinic. And that is actually illustrated by the fact, so 2018, we had one program in the clinic, soon we will have 6 programs in the clinic and 5 different compounds, all generated in-house during that almost 5-year period, which I think is quite fast. So that was one thing. The second thing is the company obviously is around for some time. Could look at this from a negative perspective. I looked at it from a positive perspective because this is really one of the remaining teams internationally with a lot of antibody experience. That means discovery, development, and manufacturing. And that's, I think, also a very, very strong aspect of the company. And number 3, what I saw, the company has actually done excellent research. So the R&D group, research and discovery, is led by Björn Frendéus. He's also actually a professor in Southampton in the oncology unit there. And they have generated a very, when I looked at it in 2018, interesting preclinical portfolio of new mode of actions within immunotherapy of cancer. Published it very well, just in the most high-ranking journals. So it was the combination at the time point, excellent science, forefront science, very good team around antibodies, and then a relatively integrated company that has all the aspects of discovering drugs and bringing them into the clinic and develop them. So, and that is still the case. So it's a super interesting company. So this portfolio we have translated now into the clinic. So we have a portfolio of currently 5 programs, soon 6 programs, 5 different compounds. And the platform that we have a very unique platform. It's called F.I.R.S.T. That is a reverse screening platform, so it works a little bit different than what other companies could do. So normally you would identify a target by any means, and then you would generate against that target compounds and test them. We do it differently, so we do it in a reverse manner. First of all, we don't screen on a specific target, but we screen on against patient material. So the company has a very close collaboration with the local hospital here in Lund, such that we receive fresh patient material on a regular basis, sometimes several times a week. And once that patient material is collected, the transfer to our labs takes 5 minutes, something like that, and then we start the screening. So we really screen against a clinically relevant material. So we know that something on this material might be interesting for therapeutic intervention. And then once we identify specific antibodies binding to the material of interest, we test that functionally. That's why we call it function first, actually, in order to see whether the targets and antibodies that we have therapeutic relevance. So we test it in a number of animal models. Once we see strong therapeutic effects in animal models, then we identify the target. So the target identification comes at the end of that process. So we start from patients, then confirm that we have therapeutic efficacy, at least in animals, and then we identify the target. So by the time we identify the target, we know it's coming from patients, so it has clinical relevance directly, and we know it works in animals. So it's already preclinically validated by that time point. And then we move to the clinic. So that's how we do the discovery process at BioInvent, and that's actually unique for the company. And that platform also has been now validated in a number of collaborations. So last year we did a deal with Exelixis, It's a big biotech company in the Bay Area. They're using our platform, and then we have already antibodies in clinical development with Daiichi, Takeda, Bayer in collaboration with the City of Hope, and Mitsubishi as well. So that's where we are at the moment. This year, based on that broad portfolio that we have, we expect a number of interesting data points. So we just had a press release today, actually, for one of our programs showing that it's safe. So it's a phase 1 dose escalation, it's safe, and we saw also our first signs of potential efficacy as a single agent, which is always interesting. At the end, of course, everything in cancer therapy will be combination, but it's always reassuring when you see also that your compound does something on itself. So that's good, and there will be further such news flow during the first half and second half of this year.
You mentioned efficacy. Could you tell me a little bit more about how it works and what you're looking to try and do?
Exactly, I can do that, and I will try to keep that general because we have currently 4 different compounds. They all have a different mode of action. First of all, I could say that out of those 4, 3 are first in class. So it's a completely new target that we are targeting, not clinically validated yet. And of course, the compound is also new. The press release that we had today, that is, maybe best in class. So we're following another one, which is Labiotech.eu. This is an oncolytic virus. But I go maybe one by one. So our lead compound is an antibody called BI-1206. And that's currently in 2 clinical development programs, one for non-Hodgkin lymphoma. And there what we do is we inhibit the important part of the resistance mechanism against anti-CD20 based therapy. In non-Hodgkin lymphoma, the main treatment modality is actually rituximab, anti-CD20. And that is the core and will remain the core for the next at least 10 years. And it works very well. It's very well tolerated. But every patient will become resistant at some time point. Every patient. And then, we have other options, but they're more toxic, not so well tolerated. And what we have with 1206, we basically block part of the resistance mechanism to rituximab. And by this, we make it work again. The best treatment options when you talk to surgeons, physicians rather, that they like for non-Hodgkin lymphoma. And so we have done dose escalation where we saw a very, very good response already during dose escalation. Just out of 15 patients, we had 4 complete responses, a couple of partial responses, and a couple of stable diseases. And the important thing really to mention there is that we had the complete responses that we saw were long-lasting. So some of which were really lasting for more than 2 years after the end of treatment. So that means there were patients that were not responding anymore to the standard of care. We give it in combination with our antibody. They become cured. That's what complete response is. And then that complete response was holding up for more than 2 years now and is still in complete response. And that's actually something what the regulators are looking after. Because that's why also I'm not mentioning at this stage overall response, even though that was good as well. But if you have 15 patients, what does it really mean? And when you look at other new treatment options that are developed in that space, they normally also, some of which, CAR-T as well as bispecific antibodies, they have also good overall response, but it weans off over time. And then you ask yourself, if you're a patient, what would you prefer? Rather take something that keeps me healthy for a long time, such that I don't need treatment anymore. And then, of course, also with good safety. That's what we have. So that's one thing. That's the most advanced. And there we have a couple of developments that we do. So we have also developed a sub-Q version of that antibody because it will be even better tolerated, and it's a more easy to handle product because you don't have to go to transfusion centers when you get the treatment. And that is currently in dose escalation, and that data will also come out soon. And then we developed that antibody also in the context of solid cancer. Solid cancers where patients have become resistant to anti-PD-1 and anti-PD-L1, which are currently the standard of care for solid cancers. But also there, at some time point, every patient will become resistant, unfortunately. And there we are a little bit more early in the development, so we're still in dose escalation, but we also have seen first signs of efficacy, which is actually quite promising. So that's our lead, BI-1206. And the target actually, I didn't mention that, I think, is Fc gamma2b, which is a very interesting inhibitory receptor on the cells of the innate immune system. So if you look at the immune system, you have the innate and you have the adaptive, and that's the innate part. And we are currently the only one targeting that one for these specific indications of solid cancer as well as liquid cancer. Then we have a second anti-Fc gamma RIIb antibody 1607. This is much earlier because there we just started clinical development last summer. It's developing quite nicely. We do that in combination with trastuzumab. It has a different mode of action. And there we're looking forward to see the first data set during the second half of this year for 1607. Then we have a program around a target which is called TNF receptor 2, which is potentially one of the new checkpoints. And there's now quite some competition. So we started this, we were almost alone. Now there's Novartis, there's Beijing, and a couple of other Chinese companies also developing antibodies around that compound, but we're still leading the pack. So we are the first ones, we're the first ones in the clinic, and we're also the most advanced in the clinic. So we have two antibodies. 1808 is our lead against that target, and there we're finishing dose escalation as a single agent. The data set should be also available soon. At the same time point, we also develop it as a combination therapy with pembrolizumab, Keytruda, and that data set will be available during the second half of this year. And then coming back to the press release that we had this morning, so this is a program called BT001, which we run in a joint venture with Transgene, and we contribute our proprietary anti-CTLA-4 antibody, which is a very interesting target. So there's already ipilimumab, which is out as a product, very efficient, but quite toxic. And we are having a nicely differentiated version of that antibody. And Transgene, our partner, is contributing the oncolytic virus platform. And basically what we have done, we've cloned the gene for anti-CTLA-4 into the oncolytic virus. And then we can infect the patient with the oncolytic virus, and then it will infect tumor cells, solid tumor cells. So we're developing in solid tumors. And then the virus will destroy those tumor cells, at the same time point also produce anti-CTLA-4. So we're combining two mode of actions there. And what we could see preclinically as well as clinically, that anti-CTLA-4 is really only expressed and produced in the solid tumor environment. And you can't see any systemic exposure. And that's exactly what we wanted. And coming back then to the press release, so what we could see in the dose escalation, so that's finished now, it's safe, and we see signs of efficacy already as a single agent, and now we move that into the second part, which is then the combination with pembrolizumab. So as you can see, there will be more or less 1, 2, 3, 4 additional data reads out this year. All early data sets, so dose escalation mainly. But that will then nicely prepare for next year, where we then at least have 2 programs in dose expansion, so then generating even later stage data.
I think one of the things that everybody talks about is cost when it comes to treatments, because obviously you want everybody that has cancer to be able to get treatment. Is that something that you can address?
Yeah, so in general, so I can't give you exact numbers for the, because that's too early. But what I can tell you, so if you look at the cancer landscape, there are CAR-Ts, all the cellular-based therapy, super expensive, because, it's a process where you have to generate the cells, you have to keep them in culture. And I can't tell you exactly, but I think it's easy to check in the net, the cost for that, but it's actually quite expensive. The other big class that kind of came out is, multi-variant antibodies, bispecifics. They're also quite expensive in production because they have lower yields in production, et cetera, et cetera. So, and what we have is, simple antibodies with a very interesting, so completely natural human antibodies with a very interesting mode of action. And they are also relatively cheap to produce compared to those 2 other elements. So I would say, yes, if we can keep the efficacy that we see, we will also have a cost advantage compared to those other 2 modalities that I've mentioned. But quite clearly will be, yeah.
That's definitely really good news because I guess ultimately it's all about the patient anyway.
Absolutely, and that's what drives us. It's about the patient, and then of course, if you can really address high unmet needs, then it also at the end will pay off for the investors. That's of course important for them. But us, Cecilia and myself, what is driving is the patient need, and then of course it has to compete with everything out there as well, which will ensure that whatever we come out with has to survive in the market and will be also efficient, right? Cost-wise as well as efficacy-wise.
And where are you going to be utilizing this? Is it going to be Europe, US?
So what we do is, so far we always have developed all our clinical programs under the regulatory in Europe, but also under IND. Also, all our programs have an IND. Which does not necessarily mean that we already develop it in the US, because during phase 1, what you want to do is generate quickly data, and if you can do that efficiently in Europe, that's what we do. If you have to go to the US in order to generate good data quickly, then we also go to the US. But all the programs that we have are approved for clinical development in Europe as well as in the US. And then I can mention that the lead program is already partnered, specifically for geography, which is China, Hong Kong, Macau, and Taiwan. And the partner there is CASI Pharmaceuticals, which is a Nasdaq-listed Chinese company. And they also have, approval in China for developing it currently in non-Hodgkin lymphoma. So there will also soon will come an update. So we were a little bit hampered during the COVID situation, especially in China. Everything was closed down, obviously. But now it's open, and CASI is really working hard and diligently to generate additional data, and that will come on top of the data that we already have disclosed and that we are generating. So for that program, we also have approval in China.
We've got a lot of work ahead, but it seems like it's going well.
Yep. So you're absolutely right. So there's a lot of work ahead, and it's going well in that sense because we have grown as a company. So when I joined, we were around 60, I think, employees roughly, and now we're a little bit more than 100. So it's not a exponential growth, but it's a steady growth, brick by brick basically. And what we really have grown is in the clinical development department, because when I joined, it was a small team consisting of a handful of people. Led by our Chief Medical Officer, Andres McAllister. And now this is probably a group around 20, 25 people, with clinical operation, regulatory, and we all built that in-house. But we need to do this because now we have 5, soon 6 programs, and then you need a significant team also in-house. We do a lot of, with CROs, obviously, but you also need a significant team in-house to make sure that you execute properly.
Okay, great. Is there anything you'd like to add that we didn't talk about?
So we have 2 targets. Fc gamma R2b is one, and then TNF receptor 2 the other one, where we have not only one antibody, one modality, but 2. And the reason for that is basically then, the target in itself becomes a platform, because you can generate different antibodies with different mode of functions that would allow to combine it in different manners in order to grab more of the space around that target. And it's quite interesting in the TNF receptor 2 space, because if we look at our competitors, they either have an antibody like our lead, BI-1808, which is a so-called blocking deleting antibody, or they have something which is more similar to what our version, BI-1910, which is still in preclinical development, but soon in clinical development, which is an agonist. So our competitors have either or, we have both. And if you ask why is that, that is back to the first platform that I described briefly at the beginning, because that platform is very versatile in the sense, once we identify a new target, then we would not sit there with just one antibody. Normally we have 10, sometimes 100, sometimes 1,000 different antibodies for one target. And they bind to different epitopes. They could be agonistic, antagonistic. And then what we do when we have this, let's say, multitude of antibodies, we screen them all one by one in different preclinical animal models in order to pick the most efficient one. And then we put those to clinical development. And I think this kind of access other companies don't have. So they would have, a system that provides them with one specific antibody at the end, or two, or maybe three, but not with hundreds and in some cases even thousands different antibodies. And that's why we can really, once we do the screening, it's very comprehensive, then we have the space in our hand and then basically pick from there what we want to develop. And I think that's a very important other or additional competitive factor that we have. Absolutely.
All right, and that's it for another week. There's no short work week in June, at least here in the UK. Not sure how we will all cope with all of that work, but at least the weather's been great. Sadly, there are lots of forest fires around the world at the moment because of all the dry weather, so hopefully those all get taken care of very soon. So thanks a lot for joining us, and I hope wherever in the world you are, you have a great week ahead. Take care, and you'll join us next week for another Beyond Biotech.